Chapter 41 Osmotic Regulation and Excretion Figure 41.1 Just as humans recycle what we can and dump the remains into landfills, our bodies use and recycle what they can and excrete the remaining waste products. Our body's complex systems have developed ways to treat waste and maintain a balanced internal environment. Credit. Modification of work by Redwin Law. Introduction. The daily intake recommendation for human water consumption is 8 to 10 glasses of water. In order to achieve a healthy balance, the human body should excrete the 8 to 10 glasses of water every day. This occurs via the processes of urination, defecation, sweating and, to a small extent, respiration. The organs and tissues of the human body are soaked in fluids that are maintained at constant temperature, pH, and solute concentration, all crucial elements of homeostasis. The solutes in body fluids are mainly mineral salts and sugars, and osmotic regulation is the process by which the mineral salts and water are kept in balance. Osmotic homeostasis is maintained despite the influence of external factors like temperature, diet, and weather conditions. Chapter Outline 41.1 Osmoregulation and Osmotic Balance 41.2 The Kidneys and Osmoregulatory Organs 41.3 Excretion Systems 41.4 Nitrogenous Wastes 41.5 Hormonal Control of Osmoregulatory Functions 41.1 Osmoregulation and Osmotic Balance By the end of this section, you will be able to do the following. Define osmosis and explain its role within molecules. Explain why osmoregulation and osmotic balance are important body functions. Describe active transport mechanisms. Explain osmolarity and the way in which it is measured. Describe osmoregulators or osmoconformers and how these tools allow animals to adapt to different environments. Osmosis is the diffusion of water across a membrane in response to osmotic pressure caused by an imbalance of molecules on either side of the membrane. Osmoregulation is the process of maintenance of salt and water balance. Osmotic balance, across membranes within the body's fluids, which are composed of water, plus electrolytes and non-electrolytes. An electrolyte is a solute that dissociates into ions when dissolved in water. A non-electrolyte, in contrast, doesn't dissociate into ions during water dissolution. Both electrolytes and non-electrolytes contribute to the osmotic balance. The body's fluids include blood plasma, the cytosol within cells, and interstitial fluid, the fluid that exists in the spaces between cells and tissues of the body. The membranes of the body, such as the pleural, serous, and cell membranes, are semi-permeable membranes. Semi-permeable membranes are permeable, or permissive, to certain types of solutes and water. Solutions on two sides of a semi-permeable membrane tend to equalize in solute concentration by movement of solutes and/or water across the membrane. As seen in Figure 41.2, a cell placed in water tends to swell due to gain of water from the hypotonic or low salt environment. A cell placed in a solution with higher salt concentration, on the other hand, tends to make the membrane shrivel up due to loss of water into the hypertonic or high salt environment. Isotonic cells have an equal concentration of solutes inside and outside the cell. This equalizes the osmotic pressure on either side of the cell membrane, which is a semi-permeable membrane. Figure 41.2 Cells placed in a hypertonic environment tend to shrink due to loss of water. In a hypotonic environment, cells tend to swell due to intake of water. The blood maintains an isotonic environment so that cells neither shrink nor swell. Credit. Mariana Ruiz Villarreal. The body does not exist in isolation. There is a constant input of water and electrolytes into the system. While osmoregulation is achieved across membranes within the body, excess electrolytes and wastes are transported to the kidneys and excreted, helping to maintain osmotic balance. 1174 Chapter 41, Osmotic Regulation and Excretion Access for free at OpenStax.org. Need for osmoregulation. Biological systems constantly interact and exchange water and nutrients with the environment by way of consumption of food and water and through excretion in the form of sweat, urine, and feces. Without a mechanism to regulate osmotic pressure, or when a disease damages this mechanism, there is a tendency to accumulate toxic waste and water, which can have dire consequences. Mammalian systems have evolved to regulate not only the overall osmotic pressure across membranes, but also specific concentrations of important electrolytes in the three major fluid compartments: blood plasma, extracellular fluid, and intracellular fluid. Since osmotic pressure is regulated by the movement of water across membranes, the volume of the fluid compartments can also change temporarily. Because blood plasma is one of the fluid components, osmotic pressures have a direct bearing on blood pressure. Transport of electrolytes across cell membranes. Electrolytes, such as sodium chloride, ionize in water, meaning that they dissociate into their component ions. In water, sodium chloride, dissociates into the sodium ion, Na+, and the chloride ion, Cl. The most important ions, whose concentrations are very closely regulated in body fluids, are the cations sodium, Na+, potassium, K+, calcium, Ca+2, magnesium, Mg+2, and the anions chloride, Cl, carbonate, CO3-2, bicarbonate, HCO3, and phosphate, PO3. Electrolytes are lost from the body during urination and perspiration. For this reason, athletes are encouraged to replace electrolytes and fluids during periods of increased activity and perspiration. Osmotic pressure is influenced by the concentration of solutes in a solution. It is directly proportional to the number of solute atoms or molecules and not dependent on the size of the solute. Molecules. Because electrolytes dissociate into their component ions, they, in essence, add more solute particles into the solution and have a greater effect on osmotic pressure per mass than compounds that do not dissociate in water, such as glucose. Water can pass through membranes by passive diffusion. If electrolyte ions could passively diffuse across membranes, it would be impossible to maintain specific concentrations of ions in each fluid compartment, therefore they require special mechanisms to cross the semi-permeable membranes in the body. This movement can be accomplished by facilitated diffusion and active transport. Facilitated diffusion requires protein-based channels for moving the solute. Active transport requires energy in the form of ATP conversion, carrier proteins, or pumps in order to move ions against the concentration gradient. Concept of osmolality and milliequivalent. In order to calculate osmotic pressure, it is necessary to understand how solute concentrations are measured. The unit for measuring solutes is the mole. 1 mole is defined as the gram molecular weight of the solute. For example, the molecular weight of sodium chloride is 58.44. Thus, 1 mole of sodium chloride weighs 58.44 grams. The molarity of a solution is the number of moles of solute per liter of solution. The molality of a solution is the number of moles of solute per kilogram of solvent. If the solvent is water, 1 kilogram of water is equal to 1 liter of water. While molarity and molality are used to express the concentration of solutions, electrolyte concentrations are usually expressed in terms of milliequivalents per liter. MEq, L. The MEq, L is equal to the ion concentration, in millimoles, multiplied by the number of electrical charges on the ion. The unit of milliequivalent takes into consideration the ions present in the solution, since electrolytes form ions in aqueous solutions, and the charge on the ions. Thus, for ions that have a charge of 1 milliequivalent is equal to 1 millimole. For ions that have a charge of 2, like calcium, 1 milliequivalent is equal to 0.5 millimoles. Another unit for the expression of electrolyte concentration is the milliosmole, MOSM, which is the number of milliequivalents of solute per kilogram of solvent. Body fluids are usually maintained within the range of 280 to 300 MOSM. Osmoregulators and osmoconformers. Persons lost at sea without any freshwater to drink are at risk of severe dehydration because the human body cannot adapt to drinking seawater, which is hypertonic in comparison to body fluids. Organisms such as goldfish that can tolerate only a relatively narrow range of salinity are referred to as stenohaline. About 90% of all bony fish are restricted to either freshwater or seawater. They are incapable of osmotic regulation in the opposite environment. It is possible, however, for a few fishes like salmon to spend part of their life in freshwater and part in seawater. Organisms like the salmon and molly that can tolerate a relatively wide range of salinity are referred to as urohaline organisms. This is possible because some fish have 41.1 osmoregulation and osmotic balance 1175 evolved osmoregulatory mechanisms to survive in all kinds of aquatic environments. When they live in freshwater, their bodies tend to take up water because the environment is relatively hypotonic, as illustrated in Figure 41.3a. In such hypotonic environments, these fish do not drink much water. Instead, they pass a lot of very dilute urine, and they achieve electrolyte balance by active transport of salts through the gills. When they move to a hypertonic marine environment, these fish start drinking seawater. They excrete the excess salts through their gills and their urine, as illustrated in Figure 41.3b. Most marine invertebrates, on the other hand, may be isotonic with seawater, osmoconformers. Their body fluid concentrations conform to changes in seawater concentration. Cartilaginous fishes' salt composition of the blood is similar to bony fishes. However, the blood of sharks contains the organic compounds urea and trimethylamine oxide, TMAO. This does not mean that their electrolyte composition is similar to that of seawater. They achieve isotonicity with the sea by storing large concentrations of urea. These animals that secrete urea are called ureatelic animals. TMAO stabilizes proteins in the presence of high urea levels, preventing the disruption of peptide bonds that would occur in other animals exposed to similar levels of urea. Sharks are cartilaginous fish with a rectal gland to secrete salt and assist in osmoregulation. Figure 41.3 fish are osmoregulators, but must use different mechanisms to survive in a. freshwater or b. saltwater environments. Credit. Modification of work by Dwayne Raver, NOAA. Career Connection Dialysis Technician. Dialysis is a medical process of removing wastes and excess water from the blood by diffusion and ultrafiltration. When kidney function fails, dialysis must be done to artificially rid the body of wastes. This is a vital process to keep patients alive. In some cases, the patients undergo artificial dialysis until they are eligible for a kidney transplant. In others who are not candidates for kidney transplants, dialysis is a lifelong necessity. Dialysis technicians typically work in hospitals and clinics. While some roles in this field include equipment development and maintenance, most dialysis technicians work in direct patient care. Their on-the-job duties, which typically occur under the 1176 Chapter 41, Osmotic Regulation and Excretion, access for free at openstax.org. Direct supervision of a registered nurse, focus on providing dialysis treatments. This can include reviewing patient history and current condition, assessing and responding to patient needs before and during treatment, and monitoring the dialysis process. Treatment may include taking and reporting a patient's vital signs and preparing solutions and equipment to ensure accurate and sterile procedures.